So it's easy to imagine that the leading strand could go a lot faster than the lagging strand. However, there are connecting proteins that will hold helicase in check. So this is going to keep it from the leading strand moving way too far ahead of the lagging strand. So you see we kind of get this, this trombone slide process where the lagging strand is kind of fed back through DNA polymerase III, which is connected over here to helicase. So in reality, it looks a little bit more like this rather than the simple replication bubble that we saw in the previous video. Once DNA has been added to a complementary strand, so we've copied the DNA, there is another set of DNA polymerases that actually go through and proofread, and this one is DNA polymerase 2. You guessed it, we were missing 2. And so this one is going to come through and proofread. If they find any that are mismatched, think about if it had found a T and instead of binding it to an A, it was accidentally bonded to a G and it didn't fit right. What will happen is it will physically be shaped different. And that is going to repair kind of typos or proofreading of the DNA molecule. So your DNA is always completely proofread before we continue on to mitosis to have replication of the cells. DNA can also be damaged. So you know that smoking and X-ray radiation can cause DNA damage. The most common type of DNA damage is called a thymine dimer. And this is when two thymines get bonded together here and they break their bonds with their complementary adenines. So this kind of DNA damage or any kind of mismatch in DNA replication gets repaired by something called nucleotide excision repair. So what happens is an enzyme here called a nuclease will come in and cut out a section of the DNA that includes a little bit before and a little bit after the damage. And then DNA polymerase will come back in and add the correct complementary DNA. And then eventually DNA ligase will come in and go ahead and bind those together. Again, notice we're going from 5' to 3' in this direction. So this is common when DNA is damaged or when it's being copied that we go through and check. So even though we check the DNA for errors, the error rate after proofreading is low, but it's not 0, so it's not insignificant. Imagine you wrote a paper for class and you had, Microsoft Word did its spell check, and then maybe you gave it to a friend to read and they spell checked it for you, and then you turn it in and then you still get, there's a grammar error or a spelling error you missed. That happens. So it happens in our DNA too. However, because of these proofreading sections, this is pretty low. Most of our DNA sequences don't actually don't cause major problems. The only time these changes become permanent is when they are happening during meiosis to form eggs and sperm to go on to the next generation. So if this happens during mitosis, you might get a mutation in your body that would change the cells in your body, but it wouldn't get passed on. The only time that this is going to cause problems for mutations that will be passed on is if this is going to be in eggs and sperm. So these changes are called mutations, and these are the only source of brand new genetic changes that happens in our genetics. So this is ultimately responsible for all of the variations that you see of life. Mutations in and of themselves are not bad or good. A mutation that causes a beneficial trait is a good mutation for that species in their environment. A mutation that causes a detrimental trait could be considered bad. Most mutations are actually neutral. They change something that's inconsequential, doesn't affect life or death in any way, and they become just neutral. So the word mutation is not inherently a bad word. Remember that eukaryotes have linear chromosomes. Prokaryotes actually have circular chromosomes, so they can start at one end and go all the way around when they replicate, and they don't have this problem with replicating their ends. However, eukaryotes, when they replicate the very last Okazaki fragment on the lagging strand, are going to have an issue. So here's the very last Okazaki fragment. This is a blunt end. This is the end of the parent strand. This is the new strand that we replicate from 5' to 3'. So DNA polymerase 1 would normally come in and remove this primer. So here we go, we've removed the primer, and now we've got DNA here. The problem is that DNA polymerase can't start here. DNA polymerase has a limitation that it cannot add DNA without having something to add onto. And it can only go in the 5' to 3' direction, so I can't go this way. The only thing I can't actually add these last 5 little DNA nucleotides. So what ends up happening at the very end of replication is the lagging strand ends up just a little bit shorter than the parent strand. So this is the new copy, and then this is the template or the parent strand. And then if we go ahead and replicate this again in the next cell, the one that had the full template strand is going to be okay. But this side, when it gets to be the template strand, so this becomes the template strand for the next round of replication, we're going to have 5' to 3' replication, and it's going to have to stop here. It's going to end up 5 nucleotides shorter than the original parent strand, which came all the way out to here. So we've essentially lost 5 nucleic acids. And this happens every time our cells replicate because they can't finish the end of the lagging strand. So these ends of our chromosomes are actually protected by sections called telomeres. So telomeres are the genes at the very end of our chromosomes, and they don't code for anything specific, but what we do is we have these long sections of code at the end of the chromosomes, and you can see them highlighted on the end of the chromosomes here, that help to protect our actual genes that we don't want to get eroded away. So they don't prevent shortening. The telomeres themselves do shorten. However, when the telomeres shorten, that means that our other genes aren't being cut away. It's like we put some extra buffer zone there. So it helps to postpone the erosion of genes. So if these genes are important, those aren't going to get eroded because we had this extra telomere section over here. They also have proteins associated to prevent cell death pathways. So if a telomere is present, that can be a way to prevent cell death because we know that this is a protected chromosome. It has been proposed that the shortening of telomeres is somehow connected to aging. We know that if the telomeres only shortened and we didn't have any way to make them longer, every generation we would have shorter and shorter chromosomes. But that's not the case. Humans have been reproducing for many, many years now, and we still have chromosomes left, so they haven't shortened away eventually. So we know when we make germ cells, so when we make gametes, eggs and sperm, there are actually enzymes called telomerase that makes telomeres longer. So when you start a new life, a baby has the longest telomeres, and then they shorten over their life. So we think that the shortening of telomeres actually protects us from cancer by limiting the number of times a cell can divide. There are specific types of cancer cells that have their own telomerase activity that allows them to continue to extend the ends of their chromosomes longer and longer, which means that they can continue to divide and divide and divide and allow those cancer cells to persist without any kind of issues to the genetics in the cancer cells. So telomerase in body cells could actually help the development of cancer. The last thing we need to look at here is kind of the larger packaging of the DNA molecule. So the DNA molecules are packed very, very tightly together. So we have on this end the loose DNA, and you can see that it continues to compact over and over. So this is a loose strand of DNA, and it gets tighter. We wrap the DNA, kind of coils around proteins here called histones. And then what we do is we take those histone complexes and we kind of wrap those around each other. And then eventually we take those and we supercoil them around each other. And then that gets condensed into these large chromosomes. So this is the most dense. When DNA is very tightly packed and it's very dense, the DNA cannot be accessed. So in order for DNA to actually be used, it needs to be loose. When DNA is dense, that's great for mitosis, for moving chromosomes around. Once the cell is in interphase and we want to actually do things with our DNA, we actually loosen it back up. So DNA can move between being tightly packed and loosely packed in specific regions where we want to access specific genes. So in a eukaryotic cell, the combination of DNA and proteins is a complex called chromatin. So that's what we say DNA is made, or chromosomes are made up of chromatin. The proteins that we wrap the DNA around are called histones. So a nucleosome is a combination of 8 histone proteins with DNA wrapped around them. And so this creates a nucleosome, which is kind of like one bead on a string. And then we take these beads and wrap them up tighter and tighter, and then loop those strings to make chromosomes. So a lot of the times when we talk about this, we will talk about it being chromatin over here when it's loose, and a chromosome over here when it's dense. Loosely packed chromatin is also called euchromatin or true chromatin, meaning that it's loose and can be used. During mitosis, we also have highly packed heterochromatin. So this is where it's going to be densely packed, and so hetero means different. This is different chromatin. When it is densely packed, it's really hard for cells to express that genetic information. So over here you can see a karyotype of 2 cells, and so these are the same chromosomes flagged. So this would be during, let's say, interphase, where this is going to be euchromatin. And you can see that each chromosome kind of creates its own little chromatin cloud, and they stay in their own little regions inside of the nucleus. So this is the nucleus. If we then take this during something like metaphase and make a karyotype, this is going to be more of heterochromatin, where it's tightly packed. So these are condensed into chromosomes. And you can see how each chromosome is flagged with its own color. So these correspond to the colors on these little kind of puff clouds over here in this image. So you can see the difference between being loosely packed in the nucleus versus densely packed chromosomes. Here are the 7 essential enzymes for DNA replication. You should take a moment and make a flashcard or do some kind of graphic organizer for each of these, learn the name, and learn the function. That should be step 1 in studying the DNA replication in this chapter. Once you know the function of all of these enzymes, then go back through DNA replication and put them in order and learn the process, how DNA copies from double helix to 2 copied helixes.